Hello and very warm welcome. Dear students, today we will be discussing the topic heterospory and seed habit. Primitive vascular land plants produced spores which were all morphologically and structurally alike, a condition called homospory. All groups of land plants up to lower pteridophytes such as club mosses and horsetails are homosporous. These plants show a life cycle like one found in ferns, in which spores are produced that germinate into small gametophytes which ultimately produce male and female gametes. The male gametes would swim across moist soils to find the female gametes produced in the female sex organs on the same or another gametophyte, where they would fuse to produce an embryo which would germinate into a sporophyte. This mode of reproduction, however, restricted early land plants to thrive only in damp environments to transport male gametes to their destination. Therefore, early land plants were forced to be confined to lowlands near shores and streams. The evolution and development of the process of heterospory freed them from this limitation. Heterospory, as the name suggests, is the production of spores by the sporophytes of land plants which are morphologically and physiologically of two types, which include microspores and megaspores. Heterospory evolved from homospory independently in several plant groups in the Devonian period as part of the process of evolution at the time of sex differentiation. Heterosporic plants produce small spores called microspores which either germinate to become male gametophytes or have reduced male gametophytes packaged within them, and large spores called megaspores that germinate into female gametophytes or have female gametophytes packaged within them. Heterospory is known in 9 genera of pteridophytes, which include Selaginella, Isoitis, Cetulitis, Marsilia, Pilularia, Reginalidium, Salvinia, Azola, and Platysoma. Some of these genera generally approach the seed habit. The principal difference between homospory and heterospory is the separation of sexes at different points in the life cycle. Nevertheless, the evolutionary implications of this difference are profound. For example, seed plants which have female gametes in ovules and male gametes in pollen grains must show some differentiation of male and female gametophytes. Furthermore, an earlier separation of the sexes in the life cycle can be regarded as a division of labor, a common theme of evolutionary advancement. A detailed study of heterosporous forms has revealed that heterospory originated due to reduction in the number of spores within sporangia. When all the spores in a sporangium are functional, there is greater competition for nutrition, and as such a spore receives a limited food supply, and consequently its size remains smaller, that is, microspores are formed. On the other hand, if some of the spore mother cells in a sporangium disintegrate during development, the remaining ones get sufficient nutrition for the development, and consequently their size is increased, that is, megaspore is formed. The above interpretation of the origin of heterospory received support from paleobotanical, developmental, and experimental studies. Let us first discuss paleobotanical evidences of heterospory. Although most of the earliest vascular plants were homosporous, many fossils of the late Devonian and early Carboniferous period, for example, Lepidocarpon, Lepidostrobus, Mesocarpon, Sigillariostrobus, Calamocarpon and Calamostychus were heterosporous. According to Scott 1894, an indication of heterospory can be seen in two species of Calamostychus, which include Calamostychus bainiana and Calamostychus cashiana. In Calamostychus bainiana, most of the sporangia were with large number of similar spores in tetrads, but in some sporangia micro and megasporangium occurred. There were some aborted spores in the megasporangia, indicating that megaspores were formed at the cost of abortion of some of the spores in the sporangium. A similar abortion of the spores was also observed in certain species of Lepidocarpon, Calamocarpon and Heteropteris. For example, a mature megasporangium of Lepidocarpon and Calamocarpon had only a single functional megaspore, as other spores aborted during development. And in Heteropteris, a megasporangium had two functional megaspores and two aborted megaspores. These examples indicate that heterospory has not evolved in living forms, but was also present in fossil plants, and it originated due to disintegration of some spores in sporangium. Now, let us discuss the evidences from developmental studies. Developmental studies in pteridophytes, particularly the events that took place during formation of sporocytes, meiosis and maturation of spores, provide a real insight into the understanding of heterospory. In Selaginella, the development of micro and megasporangium is similar till the differentiation of sporocytes. Subsequently, in the microsporangium, all the sporocytes undergo meiosis. With the result, a large number of microspores are formed. On the other hand, in the megasporangium, all sporocytes except one abort and the surviving sporocyte undergoes meiosis, forming 4 large functional megaspores. In some species of Selaginella, there is variation in the size of megaspores. For instance, in Selaginella ctenophylla, 2 megaspores of a tetrad are slightly larger than the other 2, and in Selaginella molliceps, 1 megaspore is larger and remaining 3 are smaller. An extreme case of reduction is found in Selaginella erythropus, where a megasporangium has only a single large megaspore with no traces of aborted spores. In Isoitis, the microsporangia and megasporangia are identical till the differentiation of sporogenous tissue. In the microsporangium, almost entire sporogenous tissue forms sporocytes, which after meiosis give rise to a very large number of microspores. In the megasporangium, however, a part of the sporogenous tissue and also many sporocytes degenerate, and they provide nutrition to the growing sporocytes, that is megaspores. There are only 50 to 300 megaspores in a megasporangium. In Marsilia, differences in the development of microsporangium and megasporangium do not become evident until after meiosis. In a microsporangium, all the 64 microspores formed after reduction division are functional, but in the megasporangium, though also 64 spores are formed, only 1 is functional and the rest disintegrate. A similar condition also exists in Salvinia and Azolla. Developmental studies have thus shown that determining processes of heterospory becomes operative either before or after meiosis. Before meiosis, for example, in Selaginella and Isoetis, and after meiosis, for example, in Marsilia, Salvinia and Azolla. Now, let us discuss evidence from experimental studies. Experimental studies on Selaginella and Marsilia have shown that heterospory originated due to nutritional factors. It was observed that if the photosynthetic activity of Selaginella was slowed down by keeping it in low light intensity, then only microsporangia developed. Due to low photosynthetic activity, nutrition became a limiting factor and spores could not grow in size. Thus, under such conditions, only microspores were produced. Similar results were obtained in experiments done on Marsilia. Now, let us discuss importance of heterospory. Heterospory expresses sex determining capability of the plant. In homosporous species, differentiation of sex takes place at the gametophytic stage, whereas in heterosporous species, differences in size of the spore is related to the sex of the gametophyte. That is, a microspore always gives rise to a male gametophyte and megaspore to a female gametophyte. Thus, in heterosporous forms, the sex of the gametophyte can be predicted at the spore stage. The biological significance of heterospory is that in heterosporous forms, development of gametophyte is endosporic and the nutrition for the developing gametophyte is derived from the sporophyte. Hence, the development of gametophyte is not affected by ecological factors as in case of independently growing gametophytes. Now, let us discuss the general characteristics of seed. A seed consists of an embryo, stored food and a seed coat. Heterospory, which probably has evolved independently in several lineages, ultimately lead to the transition of seed formation. The transition to seeds continued with the megaspore being placed in its sporangium while it germinates. Then the megagametophyte is contained within a waterproof integument which forms the bulk of the seed. The microgametophyte, a pollen grain which has germinated from a microspore, is employed for dispersal, only releasing its desiccation prone sperm when it reaches a receptive megagametophyte. The best example of this transition is displayed by lycopods. Fossil lycopod megaspores reaching 1 cm in diameter and surrounded by vegetative tissues are known, which even germinate into a megagametophyte in situ. However, they were not considered as true seeds because their nucleus does not completely enclose the spore. A very small slit remains, meaning that the seed is still exposed to the atmosphere. This has two consequences. Firstly, it means it is not fully resistant to desiccation, and secondly, sperms do not have to burrow to access the archegonia of the megaspore. Now, let us discuss the evolution of seed habit. The seed habit is the most complex and evolutionarily successful outcome of sexual reproduction in vascular plants. Since their first appearance in late Devonian, that is about 385 million years before present, seed plants have come to dominate almost every terrestrial ecosystem. They also encompass a greater range of habit than any other group of tracheophytes. The ecological diversity of this group generally is attributed to their reproductive system, which permits these plants to exploit habitats not accessible to most lower vascular plants. The earlier seed plants called progymnosperms emerged in the late Devonian period. Progymnosperm fossils show vegetative morphologies similar to seed plants, but not all progymnosperms had seeds or seed-like structures. Archaeopteryx species was the first modern tree, but it produced heterospores rather than seeds. The progymnosperms are regarded as the ancestors of the seed plants. Fossils of seed-bearing seed ferns, that is Lignopteropsida, exhibit a variety of seed and seed-like structures. The seed habit might have evolved once or several times during evolution. Three major evolutionary trends were important for the transition from the seed ferns to the gymnosperms, from the spores to the gymnosperm seed. The evolution from Homospory to Heterospory, and connected with this from Megasporangia with many spores to Megasporangia with just one functional megaspore. Second, the evolution of the integument, maternal tissue that protects the ovule. Third, the evolution of pollen receiving structures. This includes the transition to water independence of the pollination or fertilization process, as water is required for fertilization. The earliest seed plants with seeds or seed-like structures are Devonian seed ferns, which include Lignopteropsida and Pteridosperms. Several different types of pre-ovules or pre-ovule-like structures are known. Not all characteristics of the seed habit are evident in these pre-ovules. Runcaria, the oldest seed-bearing seed, had a small, radially symmetrical, integumented megasporangium surrounded by a cupule. The megasporangium bears an unopened distal extension protruding out the multilobed integument. This extension is assumed to be involved in wind pollination. Runcaria sheds new light on the sequence of character acquisition leading to the seeds. In general, a seed is simply a mature ovule containing an embryo. An immature ovule consists of a diploid megasporangium containing a single functional megaspore that develops into a haploid megagametophyte. The megasporangium is surrounded by diploid covering layers, the integuments, which evolve into the seed coat. An integumentary opening at the apical end is important for pollination in seed ferns and modern gymnosperms. This opening evolved into the micropyle. The late Devonian and early Carboniferous seed ferns are sometimes collectively called Lignopterids and include Alkinsia, Archaeosperma, Laginostoma, Morasnetia and Lignopteris. These earliest seed-bearing seed plants produce their pre-ovules or ovules on dichotomously branched sterile structures called cupules. Cupules are cup-like structures that partially enclose the ovule. In these early ovules, the nucellus was surrounded by integumentary tissue consisting of more or less free lobes. The integumentary lobes curved inward at their tips, forming a ring around the apical end. The integuments of the ovules evolved through gradual fusion of the integumentary lobes. The integuments later evolved into the seed coat. An opening that was left at the apical end evolved into the micropyle. It permitted pollen to enter and to fertilize the egg cell. The earliest seed plants employed hydrosperman reproduction, in which pollination occurred when wind-blown pollen was directed into a semi-closed pollen chamber. In several cases, a specialized laginostome was used for pollen capture. A laginostome is a funnel-like structure of the nucellus that projects from the top of the megasporangium. It functions as a trumpet-like pollen trapping device. From there, the pollen was delivered to the pollen chamber. A central column, that is the laginostome column, was attached to the pollen chamber floor that sealed the chamber to provide optimal conditions for pollen germination. As the megagametophytes mature, the opening to the pollen chamber is sealed and the floor is ruptured, allowing fertilization to proceed. The germinated pollen grain settles into an intimate proximity to the ovule and delivers the spermatozoids into the archegonia with the egg cells. Fossil records suggest that the sperm delivery required lysis of the megasporangium wall. The important issue was that pollination and or fertilization became more water independent during evolution, which facilitated the diversification of seed plants from Carboniferous through to the present day. So far, embryos have not been found in Devonian seed fern fossils. Dear students, till now we have studied Homospory, Heterospory, its importance and seed habit. We have studied the early Devonian and Carboniferous seed ferns and the development of seed during evolution. In our next topic, we will study the Medullosian seed ferns and the seed ferns that continued in the gymnospermous seeds that resulted in the present day angiospermous seed. Till then, goodbye.